Have you ever imagined walking alongside the giants who once dominated our planet? Can you imagine a world where creatures taller than buildings roam the lands and seas? Dear traveler, welcome. Today we're off on a breathtaking odyssey through time to discover the titans who shaped the Earth long before our time. From colossal dinosaurs and gigantic prehistoric mammals to the sea monsters that ruled the primitive oceans, let's embark on a world where size defied the imagination. But before you set off on your next adventure, remember to like the video and subscribe to the channel so you don't miss a thing. Thanks and have a great trip! To fully understand the history of the greatest creatures ever to walk the Earth, it's essential to delve into the geological timescale. This chronology, divided into eons, eras, periods, and epochs, enables us to situate the major events in our planet's history. But above all, it highlights the environments in which these giants evolved. Like an immense clock of the Earth's history, the geological timescale is divided into various key periods in the planet's evolution. These divisions enable scientists to classify the climatic, biological, and geological changes that have structured the world as we know it today. Each geological epoch corresponds to a unique environment, shaped by colossal forces. Continental drift, massive volcanic eruptions, sea level variations, and extreme climatic changes. These factors have produced conditions conducive to the emergence and dominance of certain creatures, while causing the extinction of others. Each of these giant creatures we're about to meet evolved in direct response to these specific environmental conditions. By following this geological timescale, we'll be able to understand how the forces of nature have played a considerable role, both in the size of these living creatures and in their fate. With this understanding of the geological timescale in mind, we can now delve into the origins of life on Earth. For before the giants trod the soil of our continents, life began in much more modest forms. Around 3.5 billion years ago, at a time when our planet was a hostile world dominated by primitive oceans and an oxygen-poor atmosphere, the first forms of life appeared on Earth. It was in this primitive environment, at the heart of the Precambrian era, a division that covers almost 90% of the Earth's history, that the first stages of this fascinating evolution began. At the bottom of these ancient seas, strange structures are taking shape. Resembling domes or rough carpets, they rise gently from the oceanic substratum. These sedimentary formations are the result of the sustained activity of colonies of cyanobacteria, tiny organisms commonly known as blue-green algae, capable of absorbing sunlight for photosynthesis. The surface of these structures appears stratified and marked by the undulations of superimposed layers, testifying to their age-old growth. As for their hue, it oscillates between pale green and brown, dotted with shades of green associated with the cyanobacteria that build them. In these extreme conditions, over billions of years, the Earth gave rise to one of the oldest forms of life on our planet, stromatolites. Slowly, layer by layer, stromatolites develop by capturing sediment particles. These living colonies, though frozen in time, are thus animated by the ceaseless activity of cyanobacteria, which day after day form new layers of sediment. Little by little, these isolated formations where millions of cyanobacteria gather build reefs that flourish until they dominate Precambrian marine environments. These massive structures, varying in size and shape and reaching heights of several meters, are reminiscent of modern coral reefs, but without the abundant life that characterizes them. Nevertheless, if stromatolites form an austere landscape, they are no less fascinating. For billions of years, the microscopic organisms that shaped them have thrived in shallow waters, playing a crucial role. Through photosynthesis, they enriched the Earth's atmosphere with oxygen as never before. Over time, the oxygen released by cyanobacteria accumulates, leading to the Great Oxygenation, an event that will transform the Earth's atmosphere. This created the conditions necessary for the emergence of more complex forms of existence, and life was finally able to blossom. During the Precambrian era, tiny cyanobacteria laid the foundations of the biosphere as we know it today. They demonstrate that even the most insignificant creatures can have a monumental impact on Earth's history. This is why, although isolated in ancient seas, stromatolites can be considered the first giants of Earth's history. They bear witness to the power of the smallest life forms in the evolution of our planet and its inhabitants. With the oxygen content of the Earth's atmosphere having risen to almost 20% thanks to the action of cyanobacteria, the planet is about to enter a new stage of evolution, notably with the appearance of multicellular organisms. After billions of years of life existing mainly in the form of simple cells, evolution is on the verge of a major transformation. More complex forms of life able to differentiate in terms of tissues and adopt a variety of genera are about to appear. It was at the end of the Precambrian era, around 600 million years ago, that these first multicellular organisms emerged. We can't approach this stage without mentioning the famous Ediacara fauna. Discovered in the rock formations of Australia and many other parts of the world, they represent a unique assemblage of creatures that defy our understanding of traditional life forms. These living beings, which populated the shallow seabed, are among the oldest examples of multicellular life. Their strange and diverse morphology, composed of soft bodies devoid of rigid structures, evokes flat fossil footprints left in sand. Most of them measure only a few millimeters to a few centimeters. These organisms, neither plants nor animals in the modern sense, probably feed by absorbing nutrients directly from the surrounding water. But among these countless creatures, let's turn our attention to the Charniodiscus. This creature stands out as one of the most remarkable organisms of this period. Its discovery gives us a valuable insight into the diversity and complexity of early multicellular beings. Charniodiscus has a truly unique appearance. Its structure is characterized by a flat body, generally disc or fin-shaped. Its broad, flattened body, often presented with a succession of centripetal patterns or grooves, evokes a vast field of flowers or a series of concentric circles. This morphology suggests a specific adaptation to life in a shallow marine environment, where these organisms could have attached themselves to the substrate and filtered nutrients from the water. But the most extraordinary thing about this creature is the size that some specimens display, sometimes reaching up to 1 meter or 3 feet in length. Whereas the previous era was marked by the appearance of microscopic, single-celled organisms, these creatures are distinguished by their gigantic size. As for the Charniodiscus's mode of life, this is a debate that still animates scientists. Some researchers believe that these organisms were sessile, attaching themselves to the seabed and feeding on particles suspended in the water, while others suggest that they were slow-moving. Because these creatures lacked a rigid structure and were devoid of shells or bones, the fossils studied today do not allow us to specify their habits with any certainty. However, the importance of Charniodiscus in the evolution of species is undeniable. As one of the earliest examples of complex multicellular organisms, it represents a key moment in the diversification of life on Earth. The presence of these creatures indicates that more sophisticated forms of life were beginning to emerge. Exploiting new ecological niches and laying the foundations for the subsequent evolution of more complex faunas that would dominate seas and lands in geological periods to come. Charniodiscus is therefore a witness to the passage of life from its beginnings to more diversified and elaborate forms. Its presence on the seabed in the Precambrian era is a reminder of how crucial the beginnings of multicellular life proved to be for the evolutionary dynamics of our planet. It also demonstrates the extent to which the earliest forms of life, particularly microscopic ones, were able to adapt to difficult conditions in a less than hospitable environment. By the end of the Precambrian era, the Earth had already come a long way. From a hostile, sterile world, the rapidly changing environment was able to welcome microscopic life, and the very first multicellular organisms established their roots. As oxygen continues to spread throughout the oceans and atmosphere, a veritable explosion of biodiversity is taking place. It was in the shadow of this transition that the Paleozoic era was born, marking the advent of incredibly varied and often gigantic creatures. The Cambrian, the first period of this new era, promises to be revolutionary. Life blossomed with an intensity never seen before. The seabed, hitherto dominated by primary life forms, was transformed into a veritable arena of evolutionary competition. New species armed with shells, spines, and other surprising adaptations will compete in a perpetual struggle for dominance. Without further ado, let's discover this emerging world where the strangest and most majestic creatures take their first steps, ushering in an era of marine giants that will ultimately redefine the face of the Earth. $540 million years ago, the seabed was still very different from the one we explore today. The shallow waters, bathed in diffuse light, masked the sandy bottom, dotted with microbial and algal mats gently undulating under the effect of the existing weak currents. Yet these early marine ecosystems are incredibly dynamic. Filled with forms of existence that seem to have come straight out of a strange dream, or even a nightmare for some, these oceans are like a veritable biological diversity fair. Unimaginable creatures inhabit these waters, evolving rapidly in a world where competition is fierce. The Cambrian is known as a period of great biological innovation, when nature experimented relentlessly, producing a multitude of species with diverse morphologies and survival strategies. Among the algae and the first coral reefs, an extraordinary fauna swarms, made up of fearsome predators and equally ingenious prey. At the heart of this teeming universe, one of the most fearsome predators of our time reigns supreme. Here's the animal Icarus, the Cambrian ocean giant, up to 1 meter or 3 feet long. While its elongated shape is reminiscent of an oversized but self-indulgent shrimp, its characteristics are far more terrifying. This segmented invertebrate uses its frontal appendages armed with sharp points to capture its prey, which is then crushed by its circular mouth, bristling with sharp bony fangs. This agile and fast hunter moves with a series of rhythmically undulating lateral lobes. In front of this efficient swimmer, the trilobites he loves have little chance of escaping. Not far from him, another remarkable creature stands out for its size and imposing appearance. This other giant of the Cambrian seas is called Titanosaurus gainesi. Its name aptly evokes its stature, which can reach a length of 50 centimeters or 20 inches, placing it in the category of the largest predators of its time. With its domed head protected by a broad shield, it is also distinguished by its hard, flattened carapace. This covers a large part of its body. This natural armature protects it from predators, while allowing it to burrow into the sediment for its favorite food. Moving close to the seabed, this benthic predator spends most of its time foraging in the substrate, where it exploits the abundant resources of microbial mats and sediment beds. To do this, it uses its particularly robust frontal appendages to pick up food particles or small animals hidden in the substrate. These appendages, equipped with spiny structures, are very effective at grasping and crushing food. The food is then directed into the large circular mouth on the underside of the body. During the Cambrian, another creature just as strange cohabits with these two giants. Admittedly, it's far less imposing, measuring 15 centimeters or 6 inches long, which is about twice the average size of many trilobites. But it is a singular, almost surreal creature. Meet Opabinia, a most extraordinary marine arthropod. Its 5 eyes on the top of its head give it panoramic vision. This enables it to spot prey and predators from all angles. But what makes it truly unique is the length of its proboscis, which resembles an extendable arm and ends in a pair of pincers. This extraordinary appendage enables it to search the sediments for food, which once seized is carried to its mouth on the underside of its body. Opabinia feeds mainly on small organisms and detritus picked up here and there. All these creatures evolve in a complex ecosystem where every interaction plays a crucial role in population dynamics. Prey, usually trilobites or small invertebrates, must be constantly vigilant to avoid falling into the clutches of predators such as Anomalocaris and Titanosaurus. But the latter, in turn, have to be quick and cunning to survive in this most ferocious of environments. As for Opabinia, less competitive, it avoids conflict by specializing in a different ecological niche, making do with what other predators leave behind. The Cambrian seabed is a world where the strange and the wonderful coexist, where each creature, with its unique adaptations, plays a role in the vast web of life with a capital V. The era of relentless innovation, where natural selection forged ever more complex forms of life, paved the way for the incredible diversity we know today. It's fair to say that the Cambrian oceans were not only a ruthless hunting ground, but also the cradle of many of the lineages that would shape marine life for millions of years to come. Just as the Cambrian laid the foundations for the further development of a number of strange and varied living species, the Ordovician period that follows is set to push this diversification even further. During this period, which stretches from 485 to 443 million years ago, the land is mainly arid and devoid of animal life. Vegetation was just beginning to appear, with the first plants and fungi. By contrast, the oceans, already inhabited by fearsome predators and a host of unusual creatures, saw the emergence of a profusion of marine species. These will develop new survival strategies and take over increasingly varied ecological niches. Prepare to plunge into a world where marine life has reached heights of complexity and grandeur never seen before. While the land is essentially covered by desert zones, the Ordovician seabed resembles a veritable underwater garden. Brachiopods, bryozoans, and trilobites dominate the landscape, amid microbial mats and beds of green and red algae, the main forms of marine vegetation in this period. Above all, these invertebrate organisms form the basis of a food chain that would support the emergence of veritable marine giants. Among the most impressive creatures of the Ordovician period are the nautiloid cephalopods, mollusks that gave rise to the first great ocean predators. One of the most fearsome of these is Endocerus. Its straight, elongated, cone-shaped tail can reach up to 6 meters or 20 feet. Sheltered by this shell, its segmented, flexible body enables it to move slowly through the water. Long tentacles emerge from the shell opening. Fitted with suction cups or hooks, they enable it to firmly grasp its victims, often trilobites or other small benthic invertebrates. Its powerful beak, similar to that of modern octopuses, crushes prey with ease. Although slow and heavy, Endocerus terrorizes deep-sea creatures with its size and strength. Another giant of the seas lives alongside Endocerus. Meet Cambrian. A close relative of Endocerus, its morphology is relatively similar. However, some specimens reach even more impressive dimensions, sometimes measuring up to 9 meters or 30 feet in length. Moving with imposing majesty, this creature is hard to miss. In fact, its method of stalking is that of an ambush hunter. Hidden among the seaweed, it uses its long tentacles to detect and capture its prey, which it then shreds in its mouth. But giant nautiloids weren't the only creatures raining terror in the Ordovician oceans. Eurypterids, better known as sea scorpions, represent another line of fearsome predators. Pterygotus and Megalograptus are the largest and most terrifying of these. Pterygotus, whose elongated body covered by a segmented carapace ends in a broad tail equipped with a sharp spine, can reach up to 3 meters or 10 feet in length. Its particularly well-developed front legs are equipped with massive pincers, which it uses to capture and crush its prey. Extremely powerful, these attributes make Pterygotus a formidable predator, capable of attacking large prey. What's more, its vision, provided by compound eyes, enables it to spot its prey at a distance before pouncing on them with surprising speed for a creature of its size. Like its cousin, the Megalograptus hunts in shallow coastal waters. Identical in morphology, it is distinguished by its long, spiny frontal appendages. This appearance makes it even more menacing, although it is generally smaller, reaching up to 1 meter or 3 feet in length. Highly agile, this predator is able to move quickly over the seabed. Camouflaged in the sediment, it pounces on its prey before immobilizing them with its spines, making escape almost impossible. The giant creatures of the Ordovician period, whether in open waters or more coastal environments, transform every nook and cranny of the seas into a theater of predation and survival. Their innovation adaptations mark one of the high points of marine evolution, a time when size, strength, and agility determine their survival. Let's continue our journey through time. A new stage is on the horizon, that of the Devonian. This period, often referred to as the Age of Fish, saw the appearance of the first large vertebrate predators, While the first terrestrial ecosystems began to take shape, inaugurating a new phase in the history of life on Earth, the oceans were populated by new species just as surprising. During the Devonian period, from 419 to 359 million years ago, the oceans became particularly dynamic and diverse. Coral reefs developed, creating complex environments that attracted an impressive variety of species. The waters are now dominated by armored fish, primitive sharks, and a multitude of invertebrates. On the seabed, extraordinary life forms are emerging. In shallow, rather coastal or swampy areas, prototaxis rise with confidence. The most beautiful specimens of these fungi or lichens, which exist mainly on land, are gigantic and can reach 8 meters or 26 feet in height. In their own way, these organisms influence terrestrial ecosystems as well as marine biodiversity. Food niches multiply, the evolution of species is thus boosted, and new monsters enter the living world. At the top of the food chain during this period was Dunkleosteus. This giant placoderm, measuring up to 10 meters or 33 feet long and weighing up to 4 tons, is entirely covered in thick bony plates. Its head and thorax are protected by this impenetrable armor. Its jaws, equipped with sheer sharp bone plates, are capable of crushing anything within reach, including other armored fish and even robust conch shells. Within seconds, they close with prodigious force, leaving no chance for their victims, who are then quickly crushed. Aggressive and opportunistic, Dunkleosteus is as much a hunter of large fish or cephalopods as it is of other placoderms, which it pursues in open water coral reefs. Its speed and power make it the ultimate predator in its habitat, with no real rival. Another equally imposing creature evolves alongside it. However, it contrasts sharply with the latter in its much more peaceful way of life. This is Titanichthys, a phlegmatic giant of 10 meters or 32 feet in length, whose weight is estimated at around 2 tons for the largest specimens. Unlike Dunkleosteus, this sea monster has a slender, elongated jaw, devoid of the sharp plates that characterize its predatory cousin. In fact, this colossus is a filter feeder. It feeds mainly on plankton and small organic particles floating in the water. With its mouth wide open to filter suspended food, it swims nonchalantly, reminiscent of our modern-day whales. This feeding habit enables it to exploit a very different ecological niche from that of Dunkleosteus, which is quite a good strategy, as it avoids direct competition with the large predators of the time. While Dunkleosteus regulates populations of fish and other organisms, this giant filter feeder plays a crucial role in controlling plankton levels and nutrient recycling. These interactions, among many others, contribute to the balance of marine ecosystems. These monumental creatures of the Devonian oceans, a place of striking contrasts, have forever marked the evolution of marine life. During the same period, however, changes were taking place on land, stimulated by a climate that remained warm and humid. With the movement of tectonic plates, formerly submerged lands of the still single continent rose higher and higher. Mountain ranges carved out a new landscape, as water periodically receded from the coastline. The first insects and spiders, which appeared between 395 and 380 million years ago, colonized these new terrestrial habitats, contributing to the diversification of the flora and fauna of the same period. Low spore-bearing plants developed in these high-moisture zones, and then, little by little, forests of cladoxydales, tree fern-like plants appeared. The end of the Devonian saw the appearance of immense forests of progymnosperms, including Archaeopteris. These plants, whose architecture is reminiscent of today's conifers, can reach heights of 40 meters or 130 feet. It was at this precise moment in the planet's history that evolution took a spectacular turn. The Devonian gave way to the Carboniferous, a period that stretched from 359 to 299 million years ago. Get ready to plunge into a universe where some insects are as big as hawks and arthropods dominate their ecosystems with unimaginable proportions. While the swampy waters are populated by fish, amphibians, and primitive reptiles, the skies are now invaded by flying creatures whose size is beyond comprehension. On the ground, gigantic millipedes roam the undergrowth, where life abounds under a dense canopy. It's in this lush, humid world, fed by exceptionally high levels of atmospheric oxygen, that insects reach incredible dimensions, marking a milestone in the evolution of arthropods. One of the most emblematic creatures of the Carboniferous is undoubtedly Meganeura monii, a kind of giant dragonfly with a wingspan of nearly 70 centimeters or 28 inches. Imagine an insect the size of a large crow flying nimbly over swamps. With its 1,000-faceted eyes, powerful mandibles, and intricately patterned transparent wings, Meganeura is a formidable aerial predator. Capable of capturing its prey in mid-air at impressive speed, the small insects it comes into contact with are no match for it. Another monster of the Carboniferous skies is the Meganeuropsis permiana. This member of the Meganosopterus family can reach a wingspan of 71 centimeters or 28 inches. Its elongated body and broad wings enable it to hover over the swampy forests that are home to countless prey. It can spot the slightest movement on the surface of the water or in the foliage, a sign of the presence of some small vertebrate or other insect of much more modest size, which it feasts on at will. These giant insects, often compared to dragonflies, are among the largest insects that have ever existed on Earth. But fortunately for us today, these animals, which testify to the diversity and grandeur of prehistoric life, no longer exist today. Insects of this size wouldn't just give you hives. Just imagine what your summer evenings would be like if mosquitoes were 10 to 15 times their current size. But let's go back to the Carboniferous and turn our gaze to the ground this time. Another creature makes its appearance. This is the Arthropleura, a gigantic arthropod that can reach up to 2.5 meters or 8 feet in length. It's one of the largest arthropods Earth has ever borne. Similar to a modern millipede, but of monumental size, this impressive invertebrate roams the swamps and dense forests of the Carboniferous, crushing mosses and ferns under its weight. In fact, Arthropleura feeds mainly on the abundant vegetation. Its flattened body is made up of around 30 articulated segments, each covered with protective plates. Thanks to its gigantic size and robust carapace, it has few natural predators. The Carboniferous, with its air giants in particular, represents a fascinating chapter in evolution that is still in its infancy. We've now reached the next era, the Permian, which is none other than the last period of the Paleozoic era. This period which stretches from 299 to 252 million years ago, marks a crucial transition in Earth's history. At the dawn of this period, the configuration of the continents changed radically with the formation of Pangaea, the supercontinent that alone encompasses almost all of today's landmasses. This grouping had a profound effect on the climate, creating drier and even more extreme conditions than in previous periods. The vast inland areas of Pangaea experienced scorching summers and harsh winters, while coastal regions enjoyed a more temperate climate. The lush forests of the Carboniferous gave way to more varied landscapes, including coniferous forests and savannas. Many fascinating large animals appeared during this period. These included diconodonts, large herbivores that could reach 3.5 meters or 12 feet in length. Equipped with canine tusks and resembling rhinoceroses, they were among the first warm-blooded animals. But this period was above all marked by the emergence of the first large reptiles. With their scale-covered skin and adaptability to drier conditions, they were perfectly equipped to thrive in the arid environments of the Permian. Among the iconic Permian creatures, the Dimetrodon, a member of the Pelycosaur family, is particularly noteworthy. Although often confused with dinosaurs due to its imposing size and spectacular appearance, it actually belongs to the synapsid family, an ancestral group of mammals. Measuring up to 4.5 meters or 15 feet long and weighing around 250 kilograms or 155 pounds, it is easily identified by its broad dorsal sail, a rigid structure supported by elongated vertebral spines. The crenulated teeth of this carnivore make it a terror to behold. Thus armed, this first large terrestrial predator is able to devour larger prey. At the top of the food chain, it feeds on large herbivores and amphibians. Adaphosaurus, a herbivorous synapsid that shares the same habitat, despite being 3.5 meters or 12 feet long, is unfortunately one of its favorite victims. The Permian ended in a global ecological collapse, one of the deadliest in the planet's history. The Great Permian Extinction, the largest ever recorded, led to the disappearance of almost 90% of marine species and 70% of terrestrial species. However, the Mesozoic era that followed the Paleozoic saw the emergence of the largest predators the planet has ever known, the dinosaurs. This period, often referred to as the Age of the Dinosaurs, will see the total domination of these colossal creatures, from the ocean depths to the treetops of primeval forests. Get ready, because we're about to explore a world where grandeur and ferocity are unrivaled on Earth. But let's start with the Triassic period, which stretches from 252 to 201 million years ago. As the Earth recovers from its most recent upheaval, the climate stabilizes and becomes generally warm and dry. In the wettest regions, with lush green landscapes dominated by great plains or forests of conifers, ferns and cycadophytes, food resources are abundant, providing at the same time a vast habitat. In this thick vegetation, the sound of foliage can be heard. Behold, behold, Plateosaurus. This bipedal herbivore must be between 8 and 10 meters long. With its long neck and powerful hind limbs, this dinosaur feeds on tall vegetation. It uses its claws to tear off leaves and branches, which it swallows in a single movement. Other specimens feed not far from it. Indeed, Plateosaurus lives in herds, offering protection against certain attacks. But if it must flee, Its robust body and much shorter front legs enable it to move quickly to escape predators. Herrerasaurus is one of them. This carnivorous dinosaur, one of the first, measures between 3 and 6 meters. With its sharp teeth and claws, it is a formidable hunter. Its slender body and powerful hind limbs enable it to run at great speed, while its muscular arms and claws enable it to seize and tear apart its prey. Agile and swift, stalking its prey with precision, it is a terrifying predator for any coexisting species. But the most extraordinary creatures of the Triassic period reside in the oceans. Ichthyotitan severensis is a giant ichthyosaur that can reach a length of almost 25 meters. This impressive marine reptile, often compared to an orca for its hunting habits, roams the seas in search of fish or cephalopods. It devours its prey with ease, thanks to jaws whose bones measure 2 meters or 7 feet in length. It rivals Spinosaurus, another giant ichthyosaur measuring 35 meters or 115 feet in length, which also dominates the deep waters, weighing in at 30 tons or so. Almost as big as a blue whale, this other giant creature with its streamlined body and powerful flippers is perfectly adapted to marine life. Its elongated snout enables it to capture its prey with great efficiency. An invincible predator of the Triassic seas, it hunts down fish and cephalopods, which it then devours. This marine reptile, which resembles a giant dolphin, is unquestionably the largest marine creature of the late Triassic, but also of the Jurassic, where it has found no equal. The following millennia saw the emergence of new groups of dinosaurs, marine reptiles, and pterosaurs. Continents fragmented, creating new habitats and spectacular diversification of flora and fauna. The climate, once again warmer and more humid, allowed lush forests to flourish as never before. This proved to be the ideal setting for the evolution of the prehistoric giants who were to dominate this new period, which lasted from 201 to 145 million years ago. Here we are in the Jurassic period, a time when the Earth is populated by gigantic, extraordinary creatures. The Diplodocus is probably the most emblematic dinosaur of this period. This herbivorous giant can reach a length of 30 meters or 100 feet and weigh up to 20 tons. Belonging to the Diplodocidae, a family of sauropods, it is one of the longest land animals known to man. Recognizable by its long neck and whip-like tail, it uses its comb-like teeth to pluck high leaves from trees, which it feasts on from morning to night. Located above the eyes, its nostrils allow it to breathe easily during its long meals. Its large size, massive body supported by powerful legs, and powerful tail are strong assets, enabling it to defend itself against predators like the Allosaurus. This carnivore, measuring around 12 meters or 40 feet in length and weighing around 2 tons, is probably the most ferocious of the Jurassic period. It hunts all kinds of herbivores, which it pursues with vivacity. Its powerful hind limbs and long muscular tail enable it to run at great speed, much to the chagrin of its prey. Armed with dozens of jagged, curved teeth, it easily tears off large chunks of flesh, which it feeds on bestially. Slightly shorter than the Diplodocus, but just as exceptional, is the Brachiosaurus. This other herbivorous giant of the Jurassic period can measure up to 27 meters or 89 feet in length. On the other hand, it is much heavier than the Diplodocus, weighing between 30 and 60 tons. What makes it unique, apart from its unusual morphology, are its front legs. Longer than its hind legs, they give it an upright posture. Thanks to its head, which stands 12 meters or 40 feet high, it is able to graze on the leaves of the tallest trees. All the more so as it is able to stand up on its powerful hind legs. Let's leave the verdant landscapes of the Jurassic. Giant creatures may have developed here, but this was not the most extraordinary period in the planet's history. A new, even more fantastic period is on the horizon, the Cretaceous. This period, which stretches from 145 to 66 million years ago, is not just a continuity, but a veritable surge of power. Imagine even more gigantic creatures, even more fearsome predators. As continents continue to fragment, the world changes, and with it the size of existing species reaches new heights. You're about to discover the most impressive colossi the Earth has ever born. Get ready to witness the grandiose show featuring titanic actors. Here we are in the heart of a dense forest. The wind blowing through the thick vegetation could almost lead us to believe that the atmosphere is most serene, but it's nothing of the sort. From time to time, distant roars can be heard. Tearing through this false tranquility, they remind us of the cruelty of the Cretaceous era, dominated by gigantic creatures. Between two chilling screams, heavy footsteps shake the ground. Emerging from a grove with the sound of abused foliage, a Tyrannosaurus rex announces its arrival with a terrifying roar. You're looking at a T-Rex, arguably the most iconic dinosaur of the Cretaceous period, and one of the most fearsome predators in Earth's history. Measuring up to 12 meters or 40 feet in length, and reaching a height of 4 meters or 13 feet at the hips, it can weigh between 8 and 14 tons. Its massive head, 1.5 meters or 5 feet long, features extraordinarily powerful jaws equipped with serrated teeth. Some of these can measure up to 30 centimeters or 12 inches. Designed to tear flesh and crush bone, they make the T-Rex a super predator. But beyond its frightening dentition, the T-Rex's anatomy makes it a model of murderous efficiency. Its front limbs, though short, are surprisingly powerful and end in 2 sharp claws, ideal for immobilizing and holding down its prey. Its long, robust hind limbs enable it to move quickly despite its weight. As for its long, heavy tail, it acts as a counterweight, balancing its enormous head. This attribute greatly facilitates its agility, which is rather surprising for a creature of this size. An active predator, it has no hesitation in stalking and killing large prey, alone or in groups, leaving its unfortunate victims little chance. An opportunistic scavenger, it can also feed on carcasses abandoned by other animals. Both ruthless hunter and rival, the T-Rex embodies raw power, dominating its environment and imposing its law on other dinosaurs. However, it was not the greatest creature of its time. Spinosaurus aegyptiacus was even more gigantic, measuring up to 15 meters or 50 feet in length. This behemoth, which surpasses the T-Rex in length, can weigh between 7 and 9 tons. What's immediately striking about Spinosaurus is its fabulous dorsal sail, formed by long, skin-covered spines that can measure up to 2 meters or 7 feet in height. This emblematic structure undoubtedly has several functions. As a seductive or dissuasive device, it attracts partners and intimidates potential rivals, but it may also play a role in its thermal regulation. What sets this large carnivorous dinosaur apart from its contemporaries is that its anatomy is perfectly adapted to a semi-aquatic lifestyle. In fact, its elongated skull, measuring around 1.75 meters or 6 feet, is not unlike that of modern crocodiles. Equipped with long conical jaws, they are perfect for gripping prey with force, even slippery fish. Its powerful forelimbs end in large curved claws, while its hind limbs are relatively short, compared to those of other theropods. This is probably an adaptation to its aquatic environment. Spinosaurus spends much of its time in the water, hunting giant fish and other sea creatures. A true prehistoric monster, Spinosaurus dominates both land and water, using its large dorsal sail to stabilize and camouflage itself. But do you know which is the titan among Cretaceous titans? More placid than the previous creatures, but just as fascinating, here is the Argentinosaurus, one of the largest dinosaurs ever to exist. As long as a soccer pitch and as heavy as several large trucks, this herbivore measures 40 meters or 130 feet in length and weighs between 55 and 100 tons. As you can see, with its erect neck, it can reach treetops nearly 16 meters or 53 feet high. This giant is no loner. It travels in herds, offering each individual effective protection. Few predators are bold enough to attack a member of the group. With heavy but confident steps, they travel together across the vast plains in search of vegetation. Then, tearing off whole branches with their robust teeth, they feed peacefully, or so it seems. But the survival of this species is not as obvious as it seems. The young individuals, much smaller and vulnerable to predators, are the focus of all the adults' attention. During the Cretaceous, terror reigned both on land and in the air. Creatures like the Quetzalcoatlus, Northropi, dominated the skies. This reptile is one of the most impressive representatives of the pterosaurs and one of the largest flying animals ever observed. With a wingspan of up to 12 meters or 40 feet and an estimated weight of 250 kilograms or 550 pounds, the Quetzalcoatlus is a true marvel of natural aerodynamics. Morphologically, it is characterized by its elongated skull, up to 2.5 meters or 8 feet long, equipped with a toothless beak, ideal for catching prey or searching the ground for food. Its disproportionately long forelimbs, with an elongated toe supporting the wing membranes, enable it to glide great distances, especially when relying on thermal currents. Its relatively short but robust hind limbs give it a gait reminiscent of a giant wader. This master of the air, although capable of flying great distances, feeds mainly on the ground. It uses its long neck to peck at carcasses, capture small vertebrates, and even fish in streams. What if I told you that another Cretaceous titan of the skies rivals the Quetzalcoatlus for the title of greatest flying animal of all time? Meet the Hatzegopteryx. Fortunately for many smaller creatures, this pterosaur lives on an isolated archipelago corresponding to present-day Romania. With a wingspan of 12 meters or 40 feet and a weight of around 250 kilograms or 550 pounds, it is a veritable aerial colossus, capable of dominating the ecosystem in which it evolves. Its body, though light for flying, is solidly built. Its hollow bones are nevertheless incredibly robust, enabling it to bear heavy loads, including its gigantic wings. But what particularly sets it apart from its congeners is its massive skull, nearly 3 meters or 10 feet long, one of the most robust ever observed in pterosaurs. In contrast to the slender, elongated skull of the Quetzalcoatlus, his is thick and solidly built, surmounting a stocky muscular neck. This suggests that this creature is capable of capturing and handling good-sized prey, including the small dinosaurs it feeds on. Able to crush or swallow prey whole, thanks to its powerful yet toothless jaws, this monster of the skies rivals many of the terrestrial creatures it comes into contact with. Hatzegopteryx embodies the extreme evolution of late Cretaceous pterosaurs. With its titanic size and unrivaled power, it demonstrates nature's ability to produce life forms of unprecedented grandeur, even in the most isolated environments. This Lord of the Air is, without a shadow of a doubt, one of the most fascinating examples of pterosaur evolution. But while exceptionally large creatures thrive on land and in the skies, the oceans also hide equally imposing giants. Evolving in shallow waters and dominating its ecosystem, here's the largest marine animal of the Cretaceous, Mosasaurus. At 17 meters or 56 feet in length, or perhaps even 20 meters or 66 feet, this marine reptile is a fearsome, deep-water predator. Beware of anyone who crosses its path. This monster of the seas is capable of devouring practically everything in its path. Equipped with powerful jaws armed with conical teeth, it stalks its prey. While fish, ammonites, and seabirds make up its daily menu, it doesn't hesitate to attack other marine reptiles either, using its speed and agility to capture its victims. Especially as its elongated body and powerful fin-like tail enable it to move with frightening dexterity for its prey. But as you know, despite their proven survival and development strategies, the dinosaurs' time was running out. The end of the Cretaceous was marked by their extinction in a succession of cataclysmic events. The Earth was about to enter a new era, the Cenozoic, often referred to as the Age of Mammals. This upheaval paves the way for the rise of new life forms. The era of the giants is not over. It's just changing face as immense mammals and birds are about to take over and leave their mark on Earth's history. As the giant dinosaurs disappear, despite an Earth in upheaval, a new order begins to take shape. We are entering the Paleocene and Eocene, two pivotal periods spanning 66 to 34 million years ago. In this new era, mammals became the new masters of the world, and some of them reached impressive sizes, reminiscent of the colossi of the previous era. One of the largest carnivorous land mammals of this period is probably one of these rare predators. This is the Andrewsarchus. Measuring between 5 meters or 16 feet long, with a robust body, its weight can reach 1 ton. This terrestrial mastodon has a powerful jaw with strong, sharp teeth, perfectly suited to crushing bones, tearing flesh, and shredding carcasses. Its silhouette is reminiscent of a wolf or hyena on a much larger scale. On the other hand, its skull is enormous, supported by an extremely muscular neck and shoulders. Above all, it is a fast, agile super predator, its limbs perfectly adapted to running and pursuing prey over vast territories. A solitary creature, Andrewsarchus does not hunt in packs, which in a world where competition is fierce and resources sometimes limited, reduces the risk of conflict with its fellow creatures. Another giant lurks nearby, but just as impressive, this one is of a very different nature. It rubs shoulders with many metamynodons, and Embolitheriums, primitive rhinoceroses of more modest size. In the midst of lush vegetation, a creature moves with surprising grace for its size, the Gastornis, better known as the terror bird. The Gastornis is a land bird measuring up to 2 meters or 7 feet in height. Its massive, imposing body is supported by sturdy, powerful legs adapted to life on the ground. Its wings, small and vestigial, do not allow it to fly. At most, they contribute to its balance. Although most of its movements are rather nonchalant, it is easy to imagine that it can move quickly through its forest habitat, especially in the event of an attack, running being its only means of escape. But the most striking aspect of its anatomy is certainly its beak. Massive, hooked, and hyper-powerful, it measures almost the length of its skull. Resembling the beak of a giant parrot, it is capable of generating phenomenal force, enough to crush the hardest nuts and potentially the bones of its prey. Although Gastornis is mainly herbivorous, feeding on fruit seeds, hard vegetation, and even small branches, the lack of plant resources can lead it to vary its diet somewhat. Always on the lookout for a threat or an opportunity for food, it scours the undergrowth all the time. A natural intimidator, small mammals and other birds avoid it whenever possible, recognizing its physical dominance and ability to defend its territory with ferocity. But as the climate cooled significantly, a new period began. We are now in the Oligocene, an epoch that stretches from 34 to 23 million years ago and closes the Paleogene period. New species of mammals, better adapted to cooler, more open environments, were born. New colossuses take center stage, in turn influencing the landscapes and ecosystems on which they depend. Among the giants of this era, the most impressive is undoubtedly the Paraceratherium. Imagine a creature similar to a rhinoceros, but without horns and reaching titanic proportions. Indeed, this mammal can measure up to 7.5 meters or 25 feet high at the shoulder and weigh close to 20 tons, making it one of the largest mammals ever to walk the earth. The Paraceratherium has a long neck that enables it to reach the high branches of trees. Its diet consists mainly of leaves and hard plants. Its wide jaws are capable of crushing even the toughest plants. Its huge body, supported by massive, sturdy legs, is most impressive. Despite its size, it is a relatively peaceful animal, content to roam the vast plains and sparse forests in search of food. At the top of the vegetarian food chain, it plays a major ecological role in its environment alongside other less imposing mammals. The underwater world is rich in life. Schools of colorful fish wander around majestic coral reefs. In the deeper, quieter waters, a gigantic creature swims solemnly. If its mere presence is intimidating, the danger it represents goes far beyond. Meet the Megalodon. This gigantic shark, which can reach 20 meters or 66 feet in length and weigh between 50 and 70 tons, is often considered one of the most terrifying creatures in Earth's history. As an adult, it can be around 3 times longer than a modern white shark. It is, therefore, the largest shark ever recorded, and indeed one of the most important marine predators of all time. Its name, which literally means big tooth, reveals one of its most impressive characteristics. With its sharp teeth measuring over 18 centimeters or 7 inches, this tyrant reigns over the oceans, feeding mainly on large fish and marine mammals. Its massive hydrodynamic body gives it power and speed, enabling it to move through deep water with formidable efficiency. Its imposing head is dominated by an enormous jaw, capable of opening up to a 60-degree angle. Inside this jaw lies its most fearsome arsenal. Several rows of huge triangular razor-sharp teeth, perfect for slicing flesh and crushing bone, and perpetually renewed. As for its extremely muscular, crescent-shaped tail, it enables it to achieve sudden accelerations, which can take its prey by surprise. Its hunting techniques are usually brutal, inflicting mortal wounds. But when it comes to very large prey, such as cetaceans, it knows how to use a certain strategy. First, he inflicts wounds on their fins or tails to immobilize them. He then delivers the fatal blow before feeding on the chosen morsels. As a super predator, it has no real rivals. However, it must coexist with other large predators such as giant mako sharks or certain carnivorous cetaceans, which it keeps at a distance by its superiority. On dry land, another colossus dominates the steppe. This is one of the most gigantic land creatures in history, the Indricotherium, or Paraceratherium transoriolicum. Despite its impressive size, the Indricotherium is a peaceful herbivore. It feeds mainly on foliage. This giant, which can measure up to 8 meters or 26 feet in height and weigh almost 20 tons, aided by its long neck, can easily reach the leaves of the tallest trees. This giant mammal, which looks like a hornless rhinoceros, lives in open wooded environments, sharing its habitat with a multitude of other species in complete harmony. While its imposing size protects it from many predators, it must nevertheless be vigilant in the face of possible shortages of food resources or impacting climatic changes. When the Pliocene came to an end around 2.6 million years ago, the Earth entered a new era of climatic and geological upheaval, the Quaternary. Intense glacial cycles and the reshaping of landscapes by ice and wind meant that the decline of giants of its caliber was irremediable. New titans are born, adapted to this emerging and constantly evolving world. Get ready to discover the creatures that have made their mark in this increasingly hostile and changing environment. Our first encounter takes place on a North American steppe with the majestic woolly mammoth. This colossus, clad in thick brown fur and woolly undercoat, is perfectly adapted to the rigors of the Ice Age. At 4 meters or 13 feet high at the withers, and weighing in at around 8 tons, the mammoth is one of the largest herbivores that ever lived. Its long, curved tusks, which can reach 4 meters or 13 feet in length, are not only useful for resisting predators, he also uses them to dig through the snow for buried vegetation. Every step of this creature echoes across the vast frozen expanses, marking its territory in this harsh, unforgiving world. In the dense forests of North America, another giant makes its appearance, the American mastodon. Smaller than the mammoth, it measures around 3 meters or 10 feet in height, but still weighs up to 5 tons. Unlike its woolly cousin, the American mastodon prefers woodlands, where it roams in search of leafy vegetation and branches to feed on. In the heart of the forests and plains of South America, we meet the Megatherium, a strange animal. It is the largest of the ground sloths. This giant measures up to 6 meters or 20 feet in length and weighs almost 4 tons. It moves with incredible strength, but extremely slowly. Standing on its hind legs, it can reach high branches, which it snatches up with its long, curved claws. Herbivorous above all, it is nevertheless capable of defending itself against predators, thanks in particular to its imposing size and brute strength. On the Australian continent, another titan moves slowly. This is the Diprotodon. It is the largest marsupial that has ever existed. Weighing almost 2.8 tons and reaching over 3 meters or 10 feet in length, this giant herbivore roams the plains in search of plants and foliage. Equipped with powerful jaws and large incisors, it can chew even the toughest plants. Its thick coat protects it from the elements, while its imposing stature deters predators from approaching. The Diprotodon is the epitome of quiet power, dominating its environment by its sheer presence. Now let's turn our attention to an animal of the Eurasian plains, easily recognized by its spectacular antlers. This is the Megaloceros, or great bog deer. Although it measures nearly 2 meters or 7 feet in height at the withers, its magnificent antlers, true symbols of power and domination, extend over 3.5 meters or 12 feet in span. They are mainly used in ritual combat between males to impress females. These grandiose ornaments, while commanding respect, represent a heavy burden that limits his movements in dense, food-rich forests. That's why he roams the vast plains and forest edges, where he feeds mainly on leaves and soft grasses. We can't close this period of history without mentioning a strange and fascinating creature from the same period. Meet the Glyptodon, a gigantic prehistoric armadillo that roamed the plains of South America. Weighing up to 2 tons, this colossus is protected by a solid carapace of welded bony plates, making it almost invulnerable to attack by predators. With its rounded carapace and broad body, and measuring up to 3 meters or 10 feet in length, the Glyptodon resembles a sort of living tank. Although mainly herbivorous, it is not defenseless. Its massive tail, sometimes adorned with bony spikes, can turn into a formidable weapon to repel any assailant. The Glyptodon is a perfect example of nature's diversity and ingenuity in creating robust life forms adapted to their environment. The Quaternary, the period in which we still find ourselves today, was marked by various upheavals that had major repercussions on flora and fauna. Glacial and interglacial cycles forced some species to migrate and adapt, while others became extinct forever. To conclude this magnificent journey through the ages, let's zoom in on the largest creatures we live with today. In the vast savannas of Africa, we meet one of the most fascinating and imposing birds on our planet, the ostrich. This bird can reach up to 2.80 meters or 9 feet in height, and weigh almost 150 kilograms or 330 pounds for the males. Females are slightly smaller, measuring around 1.90 meters or 6 feet, and weighing 90 kilograms or 220 pounds. Although ostriches are totally unable to fly, they make up for this with incredible running speed. Able to run at nearly 70 kilometers per hour, or 44 miles per hour, it is the fastest land bird of all. Its long, powerful two-toed legs enable it to cover great distances in search of food. Mainly herbivorous, ostriches may nevertheless consume insects or small animals to supplement their diet. Thanks to its exceptional vision, it often acts as a sentinel, alerting other animals to the presence of potential predators. During the breeding season, the male digs nests in the ground where the females lay their eggs. They then take turns incubating them, ensuring the protection and survival of the future ostriches. Adapted to arid and semi-arid environments, the ostrich has thrived in the African savannas thanks to its unique abilities and social behavior. Not far away, still in this savanna landscape, we come across the largest of land mammals, Loxodonta africana, more commonly known as the African elephant. Males can reach up to 7.5 meters or 25 feet in length and weigh almost 8 tons, while females are no more than 6 meters or 20 feet for 4.5 tons. The African elephant is perfectly adapted to its environment. Its large fan-shaped ears help it to regulate its temperature, and its impressive tusks, which can measure up to 3 meters or 10 feet, keep predators at bay. Its trunk, a multifunctional organ, enables it to drink and feed, but also to grasp branches or anything else. African elephants live in matriarchal herds, led by an older female. Consuming up to 300 kilograms or 660 pounds of vegetation a day and drinking 200 liters of water, they have to travel vast distances to find food. But above all, they demonstrate a complex social life and an ability to feel emotions. This is why, beyond their colossal size, African elephants are truly fascinating creatures. And now to the tropical waters of Southeast Asia and Australia. This is the home of a huge marine crocodile. This is the fearsome Crocodylus porosus, the largest reptile currently living on Earth. A male specimen can reach up to 7 meters or 23 feet in length and weigh over a ton. With its thick, scaly skin, powerful jaws, and sharp teeth, the marine crocodile is an unrivaled predator. Able to swim in both salt and freshwater, it is often found in estuaries, mangroves, and coastal marshes. This opportunistic hunter feeds shamelessly on fish, mammals, or even birds caught by surprise. Its ability to remain motionless for long periods, combined with its explosive speed, makes it a most formidable predator. While it reigns supreme in its environment, it also plays a crucial role in its ecosystem. In fact, despite its fearsome reputation, it regulates the populations of its prey, thus contributing to the equilibrium of these aquatic habitats. Let's end our exploration of giant creatures by diving into the mysterious depths of the Antarctic Ocean. Let's take a look at the colossal squid, or Mesonychotothis hamiltoni, which is none other than the largest of our planet's current invertebrates. The colossal squid is a veritable giant of the cold, dark abyss. An adult specimen can measure up to 14 meters or 46 feet in length and weigh almost 500 kilograms or 1,100 pounds. With its pinkish-red skin, dotted with the chromatophores that enable it to change color, it practices the art of camouflage, probably one of its greatest assets. With its massive mantle and powerful tentacles, the colossal squid is perfectly adapted to life in the depths. Its eyes, among the largest in the animal kingdom, can measure up to 27 centimeters or 11 inches in diameter, enabling it to detect the slightest movement despite the total darkness that surrounds it. Its tentacles, equipped with suction cups and hooks, enable it to firmly grasp its prey, i.e. fish or other squid, and bring them to its powerful beak. Its impressive size and unique adaptations make it one of the ocean's most mysterious giants. Now let's meet the largest creature on our planet, the blue whale, or Balenoptera musculus. At 30 meters or 100 feet long and weighing 190 tons, the blue whale is the largest animal of all time. Its long, slender body and smooth skin attest to its perfect adaptation to marine life. Swimming gracefully in the ocean, mouth wide open, this behemoth constantly filters the water to capture krill, a small crustacean. To meet its needs, it must consume no less than 4 tons a day. Despite its titanic size, the blue whale remains a majestic and peaceful animal. By tracing the history of life on Earth, we've discovered the diversity and splendor of the greatest creatures that have populated our planet. This plunge into the past has taught us that what seems colossal at one time may seem modest at another. It has also shown us that the size and importance of living beings are always relative to their time. But beyond their impressive size, these creatures, whether terrestrial or marine, embody the infinite richness of life on Earth. A remarkable example of the evolution of the vision system can be seen in the developmental changes observed in the eyes of lampreys. These changes support the hypothesis that the lamprey's ancestors succeeded those of the hagfish in the course of evolution. Indeed, the Amocoetes, or lamprey larvae, have eye characteristics similar to those of the hagfish. In the larval stage, its eyes are small, sunken under the skin, and have a relatively undifferentiated retina, giving it very limited or non-existent vision. Over time, however, a slow process of neuronal differentiation takes place in a specific order. Ganglion cells, followed by amacrine and horizontal cells, then photoreceptors, and finally bipolar cells are formed. This sequence of differentiation is similar to that observed in jawed vertebrates for the development of their retina. When metamorphosis occurs over hundreds of thousands of years, the lamprey's eye undergoes significant transformations. It enlarges considerably, its retina differentiates completely, the lens develops, the cornea divides into scleral and dermal layers, allowing the eye to move relative to the epidermis. As in jawed fish, extraocular muscles develop, and finally, the eye bursts through the surface to form a visual organ similar to that of vertebrates. The process of eye development in lampreys suggests that these animals have inherited their visual apparatus from a common ancestor they share with hagfish. The existence of a hagfish-like larval eye, which transformed into a vertebrate-like eye in adulthood, supports the idea that lampreys evolved from a common ancestor with hagfish. The next step in the evolution of the eye was the fusion of these photoreceptors to form an eye spot. This advance enabled the organism to detect the intensity of light. In simple terms, if several photoreceptors were activated in an eye spot, this meant that there was strong luminosity and vice versa. This adaptation improved the perception of surrounding brightness. Once again, scientists have discovered clues in embryonic development. Early in the process, a neural structure develops and gives rise to the eye, forming 2 sacs, or vesicles on either side. Later, each vesicle coils to form a C-shaped retina, which lines the inside of the eye. It's very likely that evolution followed a similar pattern. At a later stage in embryological development, as the retina folds inwards, the lens forms, leading to a thickening of the outer surface of the embryo, known as the ectoderm. The ectoderm swells into the curved space created by the C-shaped retina, gradually detaching from the rest of the ectoderm to become an independent floating element. It is likely that a similar sequence of changes took place in the course of evolution. The exact date of this change remains unknown, but in 1994, researchers at Lund University in Sweden demonstrated that the optical components of the eye could evolve relatively rapidly, within the space of 1 million years. If this is the case, it is conceivable that the image-forming eye emerged from the non-visual proto-eye in a short period of time on the geological scale. In parallel with the emergence of the eye spot, organisms also developed locomotion mechanisms such as flagella and cilia. These developments led to the need to localize the light source. To fulfill this role, several eye patches fused together to form an eye patch, like a patchwork quilt. These eye patches enabled the organism to roughly locate the light source and orient itself in its direction. By grouping several eye patches together in this way, organisms were able to improve their ability to perceive and move in relation to ambient light. In order to improve localization, the eye patch had to undergo progressive expansion. However, space was an evolutionary constraint. In response to this challenge, nature came up with a solution: the eye cup, also known as the optical cup. The development of the eye cup in vertebrates proceeds as follows. The eyes take shape from evaginations of the developing diencephalon. Depressions known as optic grooves form on either side of the rostral neural plate. As the neural plate folds upwards and inwards to form the neural tube, these growing regions project outwards and are called optic vesicles. When the expanding optic vesicle comes into contact with the surface ectoderm in a specific region called the lens placode, this induces changes in both tissues. The optic vesicle then invaginates to form the optic cup, while the ectoderm undergoes differentiation to form the lens hollow, which later transforms into the lens itself. It is sometimes suggested that the invagination of the optic cup results from the growth of the lens, but observations show that this cannot be the case. In fact, a normal ocular cup and retina can develop in the absence of a lens, indicating that these developmental processes are independent and follow distinct pathways. This adaptation enabled the organism to precisely determine the angle of the incident light beam according to the photoreceptor activated. Thus, the optic cup played an important role in improving the ability to localize the light source, despite the spatial constraints faced by the organism. By this stage, the primitive eye was fully formed, and all the species that possessed this organ entered into fierce competition for available resources. This competition gave rise to a major evolutionary event, known as the Cambrian explosion, which occurred around 540 million years ago. Scientists argue that the evolution of the eye accelerated rapidly during this period, as precise detection of prey and light gave organisms a survival advantage. So the better the eye, the better the organism's ability to survive. This selective competition for improved visual systems played a key role in the rise in diversity and complexity of animal species during the Cambrian explosion. After the Cambrian explosion, the next period saw the evolution of two essential eye structures: the iris, followed by the lens. The first step was to isolate the eye from the outside environment, which was achieved by the development of a transparent, epithelial layer, now known as the cornea. Once the light detection system had been separated, the next evolutionary advantage was the ability to focus. Increased focusing power dramatically improved object localization. The first structure to develop this enhanced focusing power was the iris. Thanks to this adaptation, all incoming light was channeled into a single beam, according to the direction of the light rays, greatly improving the accuracy of light source localization. The next major development was the crystalline lens. This lens allowed light rays traveling long distances to focus directly on the retina rather than behind it. This enabled the organism not only to locate the light source precisely, but also to perceive the shape, depth, and other properties of surrounding objects in greater detail. The lens thus opened up new sensory perspectives for organisms with advanced visual systems. With the advent of the lens to capture light and focus images, the eye acquired a greatly enhanced capacity to gather information. This improvement would have generated selective pressures favoring the emergence of more efficient signal processing in the retina, going beyond the simple connections between photoreceptors and output neurons. To meet this requirement, evolution has modified the cell maturation process so that certain developing cells, instead of becoming ciliary photoreceptors, become retinal bipolar cells, which intercalate between the photoreceptor layer and the output neuron layer. This is why retinal bipolar cells bear a strong resemblance to rod and cone cells, although they lack rhodopsin and are not sensitive to light. Instead, they react to the chemical or neurotransmitter released by the photoreceptors, enabling them to play an essential role in processing visual signals before passing them on to the output neurons. Camera-type eyes offer a wide field of vision, usually around 180 degrees. In practice, however, our brains are limited in their ability to process all the visual information available at any one time, due to the limited number of nerve fibers connecting our eyes to our brains. As a result, we can only sample a fraction of the visual data available. It is likely that the first camera-like eyes faced an even more severe limitation, with probably an even smaller number of nerve fibers. This constraint would have exerted considerable selective pressure in favor of the evolution of muscles to move the eyes. The presence of such muscles must have existed around 500 million years ago, as the arrangement of these muscles in lampreys, whose lineage goes back that far, is almost identical to that of jawed vertebrates, including us humans. Despite the ingenious features of vertebrate eye evolution, a number of rather counterproductive traits can be identified. For example, the retina is positioned upside down, which means that light has to pass through the entire thickness of the retina, including nerve fibers and intermediate cell bodies that scatter light and impair image quality before reaching the light-sensitive photoreceptors. In addition, blood vessels cover the inner surface of the retina, casting unwanted shadows on the photoreceptor layer. Another tricky aspect is the presence of a blind spot, where nerve fibers that cross the surface of the retina come together before tunneling through it to emerge as the optic nerve at the back of the eye. This list of inelegant features could be extended to include other examples. These imperfections are by no means inevitable features of a camera eye, as octopuses and squids have evolved independently of camera eyes which do not have these defects. Examining the vertebrate eye in an evolutionary context highlights these seemingly absurd shortcomings as the consequences of an ancient sequence of evolutionary steps, each of which brought advantages to our vertebrate ancestors, long before they developed advanced vision. The eye has evolved in many different ways over time, giving rise to a variety of features in different species. Jellyfish, for example, have eyes, and although they have no brain, these eyes transmit information directly to the muscles for rapid reaction. Nautiluses have an aperture without a crystalline lens, adopting a different focusing mechanism. Flies, on the other hand, have many eyes, giving them a wider field of vision. The diversity of ocular adaptations continues with predators, which have forward-facing eyes for better depth perception, while prey have lateral eyes, offering a wide field of vision for detecting potential dangers. In the case of humans, they have developed the ability to artificially correct their vision using devices such as spectacles, enabling them to compensate for certain visual imperfections. Ultimately, the list of ocular adaptations is truly infinite, with each species evolving uniquely to suit its environment and specific needs. All the auditory organs of today's terrestrial vertebrates, whether reptiles, birds, or mammals, have evolved in completely distinct ways. Ciliated cells, the sensory cells at the base of the auditory organs of all vertebrates, emerged very early in the evolutionary history of animals. They play a significant role in today's non-vertebrate animals, such as ascidians, and can be traced back to the first animals with well-developed tissues, the cnidarians, like sea anemones. The auditory part of the ear evolved from a vestibular organ whose evolutionary origin, in particular the sensory cells themselves, resembles that of the lateral line, a body surface organ used by fish to detect the movement of fluids in water. The lateral line has sensory hair cells similar to those in the cochlea, whose responses are strongly influenced by viscous damping and are linked by a gel-like structure. The damping problem was solved early on by evolving the active mechanisms of the sensory cells and linking them via a gel-like structure to increase both their sensitivity and frequency selectivity. These two features have survived 500 million years of evolution and are common to vertebrate inner ears. During the Carboniferous period, from -359 to -299 million years ago, tetrapod ancestors successfully migrated to terrestrial life, paving the way for further adaptations of existing sensory systems, including hearing, from aquatic to terrestrial life. Thus, over hundreds of millions of years, progressive adjustments were made to these organisms so that they could adapt to higher frequency stimuli, first in an aquatic environment and then in an aerial one. It is assumed that the ancestors of amniote vertebrates had a limited hearing capacity for loud, low-frequency sounds, as they lacked a tympanic middle ear. The first terrestrial vertebrates had an auditory papilla that rested on solid tissue and was covered by an otolith, a small mineral structure present in the inner ear of certain animals, notably fish and reptiles, responsible for detecting movement and balance, and composed of calcium carbonate crystals. In amphibians, the otolith membrane was replaced by a tectorial lining that contained no otoliths. In all subsequent lineages, the basilar membrane appeared, freely suspended. In other lines, the basilar membrane showed some adaptation, with moderate frequency selectivity. Around 100 million years after the separation of the tetrapod lineages during the Triassic period, at least 5 independent developments of tympanic middle ears were observed. These developments functioned as essential impedance matching mechanisms for the efficient detection of airborne sounds. The absence of an effective middle ear prevents the detection of high-frequency sounds. This gave rise to parallel evolutions, leading to major differences in the auditory systems of tetrapod vertebrates between different clades, while ultimately fulfilling the same function: detecting, transmitting, transducing, and encoding airborne sounds. Paleontological, morphological, and developmental evidence suggests that lepidopsaurs, such as lizards and snakes, as well as archosaurs, which include birds and crocodiles, independently developed a middle ear, composed of a single ossicle. Firstly, in birds and crocodilians, the structure of the auditory organ shows marked similarities, such as the presence and distribution of similar cell types. It is therefore reasonable to assume that this structural configuration was reached or initiated at the latest around the separation of their ancestral lineages some 160 million years ago. Similar reasoning applies to lepidopsaurs. In Tuataris, which separated early from other lepidopsaur lineages, the cellular configuration of the auditory papilla is similar than in lizards. In Tuataras, as in turtles, there is only one type of cylated cell, and all cylated cells have the same orientation. Thus, the more complex structure typically seen in lizards seems to have arisen after the Tuatara lineage separated from the squamates. However, it is not known whether lepidopsaurs, including the Tuataris, developed a tympanic middle ear all at once. Finally, in mammals, the characteristic structure of the auditory organ, known as the organ of Corti, is essentially found in all recent groups of mammals, although there are marked differences between monotremes and therian mammals. We can therefore assume that the organ of Corti appeared before these lineages split, between 220 and 150 million years ago. This comparative analysis suggests that the evolution of the tympanic middle ear in each lineage had significant implications for the later development of the auditory organs of the inner ear. The reduction in bone size and the emergence of new configurations to form the tympanic middle ear probably improved not only auditory transmission in general, but especially that of higher frequencies. This new feature undoubtedly exerted additional selection pressures on the auditory organ to enable optimal transduction of information and its transmission to the brain. Over time, the mammalian cochlea evolved specifically in different subgroups. Over the course of several subsequent geological periods, mammals diverged considerably, and although the basic structure of their cochlea was relatively constant, differences in length and various specializations emerged. Not only did cochlea develop huge differences in length, they also developed specific specializations in groups pursuing different strategies for locating prey, using ultrasonic frequencies in the air, such as bats, and high ultrasonic frequencies in the water, such as toothed whales. For example, some bat species have developed cochlear regions where the length of the organ of corti dedicated to specific narrow frequency ranges is considerably enlarged. These adaptations have enabled bats to better detect and localize sounds in their environment, improving their hunting and navigational abilities. On the other hand, there is no reason to expect mammalian auditory organs to be necessarily superior or more sophisticated than those of other groups. Each present-day vertebrate group has developed an appropriate auditory solution according to the evolutionary pressures dictated by the environment, predator-prey relationships, and intraspecific communication needs. Acute sensitivity and precision in frequency resolution, characteristics commonly associated with mammalian auditory organs, have evolved independently in all groups of modern terrestrial vertebrates, in response to their own specific evolution and adaptation. Indeed, some species of lizard possess auditory nerve fibers tuned to some of the finest frequencies of any vertebrate, including most mammals. Birds of prey, particularly owls such as the barn owl, rely almost exclusively on hearing to locate their prey, so auditory sensitivity exceeds that of most mammalian species. Echolocating mammals such as bats and dolphins exhibit a wide range of hearing frequencies, a characteristic almost equaled by certain species of frog, which use ultrasonic calls to communicate. In humans, although generation times are 10 times longer than in fish, the issue is not so much the creation of hundreds of new species, but rather the adaptation of a sensory organ to optimize the processing of communication signals vital for survival and hence reproduction. Whether the evolution of the human cochlea paralleled the development of language is a fascinating question that could be explored by future research. All living cells are irritable to certain chemicals, and this predisposition of cells to be perturbed by chemicals is likely to have led to the eventual evolution of specific receptor proteins to detect chemical signals, and ultimately to specific chemosensory organs and systems such as the sense of smell. Olfactory capacity, used for chemical communication, is widespread in the majority of living beings belonging to the animal kingdom, enabling the perception and identification of chemical signals. The sensitivity and chemical range of animals' olfactory systems are remarkable, giving them the ability to detect and distinguish between thousands of different odor molecules. In the animal world, olfaction has evolved to enable the detection of signals from the chemical environment, which contains decisive information on the direction to follow, food sources, the right time to reproduce, as well as stimuli to consider pleasant or dangerous. An animal's reaction to chemical signals is the result of a combination of mechanisms acquired during its lifetime and innate predispositions resulting from the cumulative impact of individual experience and evolution through the generations on the olfactory nervous system. Evolution has shaped the olfactory system in response to two challenges: the need to explore a wide range of chemical information and the specific sensory challenges faced by each animal species. Generally speaking, vertebrates develop an olfactory system that detects odorants and pheromones through their interaction with cell surface receptors specialized on olfactory sensory neurons and organized in dramatic and subtle ways, enabling adaptations in response to major changes in environment and lifestyle, such as the transition from ocean to land and the predominant development of vision. In terrestrial vertebrates and insects, we often think of olfaction as a chemosensory modality dedicated to the detection of low concentrations of airborne volatile chemicals. Yet aquatic fish and crustaceans, while not encountering airborne volatile odorants, possess sensory systems anatomically similar to the olfactory systems of terrestrial animals. For these aquatic animals, odors are sapid molecules in solution. Olfaction is therefore not necessarily the detection of volatile molecules in the air. In this context, it has been suggested that during the transition between aquatic and terrestrial environments, new demands emerge for chemosensory systems linked to odor detection. This happened in a remarkable way, as olfactory stimuli evolved from hydrophilic to predominantly hydrophobic and volatile compounds. This transition granted terrestrial vertebrates an evolutionary option to detect volatile water-insoluble odorants. As a result, it is thought that the complex biosynthetic pathways required to produce volatile compounds, as well as the highly complex combinatorial coding that characterizes olfactory perceptions, driven by olfactory receptors, emerged suddenly in just a few 100,000 years. Comparative genomic and phylogenetic studies argue that chemosensory receptors were conserved across aquatic and terrestrial taxa, and that mammalian-like olfactory receptor genes evolved in marine environments before the first vertebrates appeared. These changes in olfactory receptors at protein level, altering their interaction with odorant molecules, play a key role in the evolution of olfactory perception. However, researchers have discovered that this adaptive evolution of olfactory perception can also be influenced by co-expressed olfactory receptors undergoing changes in copy number and protein sequence. Selection also acts on individual olfactory receptor families or subfamilies, favoring specialized adaptation or facilitating chemical communication between members of the same species. Finally, genetic variation among the thousands of unique olfactory receptor genes can lead to behavioral modifications in response to specific chemical signals. Thus, the combination of these mechanisms enables vertebrates to adapt in precise and complex ways to their changing chemical environment. An animal's genome evolves in such a way as to be closely linked to the specific tasks resolved by its nervous system, enabling it to detect odors and respond appropriately to promote survival and reproduction. Recent studies have revealed the presence of ancestral versions of synaptic genes involved in fundamental neuronal communication in various organisms without a nervous system, suggesting a progressive evolution of olfaction. In parallel, studies have clarified that the olfactory capacities of multicellular organisms, as well as chemosensory learning abilities, evolve from life forms not equipped with complex neural networks. In particular, prokaryotes have been proposed as model systems for understanding the evolution of olfactory systems in higher organisms. In fact, bacteria respond to volatile chemical gradients, but also use contact-dependent signaling. In addition, a unicellular but multinucleate eukaryotic organism, Viscerum polycephalum, has been shown to be capable of chemosensory detection. Studies have shown that olfactory receptor cells are primary bipolar neurons with a similar morphology between species. They have filamentous dendrites to increase the surface area for stimulus capture, and an axon that extends to the central nervous system. Dendritic processes are mainly of ciliary origin in most animals. Although the overall morphology is conserved, the fine details of olfactory receptor cell morphology can be adaptive, i.e., they can vary in a habitat-dependent rather than species-dependent manner. For example, the olfactory receptor cells of land-dwelling crustaceans, such as the giant thief crab, have a morphology closer to that of insects than to that of marine crustaceans. Crustaceans share a common ancestor with insects, and as they lack olfactory receptors, it has been proposed that these receptors evolved when prehistoric insects left the sea to live on land. According to this idea, olfactory receptors evolved because these ancestors must have been able to detect odorous molecules floating in the air, rather than dissolved in water. But a recent discovery contradicts this hypothesis. The discovery of a new type of olfactory receptor, ionotropic receptors, IR, has changed the order of things and opened up new avenues of inquiry into the evolution of olfactory receptors throughout the history of life. These new olfactory receptors are present in crustaceans, in which no classical olfactory receptor had been found until now. Since crustaceans were the first to appear in the history of life, before insects and before vertebrates, we can assume that these IR were inherited long before the appearance of the first insects. During development, the olfactory system takes shape from the olfactory placodes, specialized regions of the anterior ectoderm that share cellular and molecular characteristics with other placodes involved in the development of various cranial senses. Certain cell types of the olfactory system preceded the appearance of vertebrates, as did certain mechanisms involved in placode formation. It is likely that these two elements were already interconnected in the common ancestor of vertebrates and tunicates. In vertebrate ancestors, this system evolved into a more complex organ system, integrating additional tissues and specific morphogenetic processes to develop distinct olfactory components. Subsequently, the ancestral placode divided to give rise to the paired olfactory organs characteristic of most modern vertebrates. Thus, a major olfactory adaptation occurred along the amphibian and terrestrial branches of the vertebrate tree, resulting in the formation of distinct neuroanatomical structures expressing more recently derived olfactory receptors. Aquatic vertebrates such as fish and amphibians have an olfactory system similar to that of terrestrial vertebrates, but specialize for the detection of chemicals present in water rather than air. Insects, on the other hand, have a well-defined system in their antennae for detecting airborne chemicals, generally referred to as the olfactory system. However, this system is evolving convergently, independently of vertebrates to perform this function. Unlike fish, amphibians have developed a separate, vomeronasal organ, which specifically detects attractive sex pheromones specific to amphibians. In addition, they possess an air-filled main olfactory epithelium, which expresses a vast repertoire of novel Class II olfactory receptors that have acquired a role in innate reproductive behaviors. These observations suggest that odor interpretation depends not only on the odorant molecules detected, or ligands, but also on the developmental identities and specialized sensory pathways of the cells that detect these odors. Recent evidence shows that individual mammalian olfactory receptor cells can be activated by several different odorants, and that individual odorants activate several receptor cells expressing different receptor proteins. This combinatorial coding is also present in insects and fish, suggesting that it has been conserved in the evolution of olfaction. Consequently, there are striking similarities between species in the organization of the olfactory pathway, from the nature of olfactory receptor proteins to the organization of the olfactory central nervous system. Although there is a striking evolutionary convergence towards a conserved organization of signaling pathways in vertebrate and invertebrate olfactory systems, the receptor gene families involved have evolved independently. These common organizational features span a wide phylogenetic range of animals. Such conservation also implies that there is an optimal solution to the problem of odor detection and discrimination. Either this solution evolved relatively early and was then retained in evolution, or more likely, animals convergently developed identical or similar solutions to the odor detection and recognition problem. The sense of taste is a specialized chemosensory system, one of the main mechanisms used by animals to assess the nutritional quality of food, helping them to make decisions about consuming beneficial foods while avoiding harmful substances in their daily lives. Unlike touch, vision, hearing, or olfaction, which are called upon in different behavioral situations, the sense of taste has evolved to play a primordial role in food regulation and motivation. Taste systems detect essential nutrients, as well as harmful substances in food, and trigger innate behaviors that lead to the acceptance or rejection of potential food sources. Most vertebrates can perceive 5 basic taste modalities: bitter, umami, sweet, salty, and acidic. All vertebrates have some ability to avoid harmful chemicals and seek out nutrients. It is thought that each sense of taste has evolved to detect and motivate the consumption of essential nutrients, and also to detect and avoid potential poisons. It is widely accepted that sweet taste evolved in plant-eating animals to detect energy-rich simple sugars, such as glucose, fructose, and sucrose. Bitter taste, on the other hand, probably functions to ensure that an animal avoids poisons. Indeed, most poisons are bitter, and most bitter substances are harmful, although this relationship is not perfect. The salty taste allows the detection of sodium, an absolutely essential mineral. When certain species of animal lack sodium, and this usually occurs in herbivorous animals, a powerful appetite for salty taste is aroused, and for many species, salt is consumed even when there is no apparent need. As for the sour taste, many have suggested that it is involved in detecting fruit ripeness. Finally, the fifth basic taste, umami, probably serves to signal amino acids and proteins. By comparing animals from different branches of the evolutionary tree, researchers have deduced that the sense of taste probably evolved over 500 million years ago. This would have occurred before the divergence of terrestrial vertebrates, bony fish, sharks, and lampreys, when their common ancestor, a primitive fish, developed a new type of cell. These chemosensory taste receptor cells, located in taste buds, are able to detect chemical stimuli and send signals to the brain via the taste nerves to generate taste sensations. Taste buds are the gustatory end organs in vertebrates ranging from lampreys to mammals. These end organs respond to a variety of sapid chemicals and transmit signals to afferent nerve fibers from three cranial ganglia: facial, glossopharyngeal, and vagus. Over time, taste buds have undergone several modifications to adapt to the dietary needs of different animal species, which explains their varied location in these animals. Not only are they found in the mouth and pharynx, but they can also be widely distributed across the surface of their bodies. Evidence suggests that umami receptors were the first to develop. Researchers have reported the discovery of genes similar to those encoding the receptors used by humans to detect the amino acid glutamate in the genome of the elephant shark, a species that split off from other fish 400 million years ago. Sharks lack bitter taste receptors, suggesting that these genes evolved more recently. Ancient fish were probably the first vertebrate animals capable of detecting sour taste. It is likely that these fish did not taste food with their mouths, but rather could detect acidity in the ocean using the outside of their bodies. Variations in the carbon dioxide dissolved in water can create different levels of acidity, which can be dangerous for fish. So the ability to sense acidity would have been essential for their survival. Bitter taste is perhaps the taste that most intrigues evolutionary biologists. It's the Darwin's finch of taste, engineered and customized to suit a species' ecological niche. Humans have 24 or 25 different bitter receptors, depending on the individual, each recognizing unique combinations of chemicals. Toxic bitter compounds come in all shapes and sizes, so it makes sense that the receptors that recognize them are diverse. The evolution of bitter taste, therefore, also involves distinguishing between different chemicals. The diets of humans and their ancestors are mainly based on plants, which are an abundant and accessible source of nutrition. However, plants have developed physical, reproductive, and chemical defenses to protect themselves from herbivores. Herbivores must therefore contend with various obstacles such as thorns, shells, and symbioses with defending animals. Herbivores have developed various strategies to overcome the toxic defense mechanisms of plants. Among these, a particularly effective and widespread method in vertebrate animals is the detection of bitter flavors. It has long been widely recognized that many plant toxins are perceived as bitter, creating a powerful mechanism for minimizing exposure to these harmful substances. By signaling the presence of toxins in foods before they are fully consumed, bitter taste detection helps avoid their ingestion. This ability plays an essential role in the survival of herbivores, helping them to identify and avoid potentially dangerous plants. The ubiquity of bitter perception in vertebrates and its complex role in humans have raised questions about its evolution. One of the most remarkable revelations of vertebrate comparisons is that bitter taste receptors are a relatively recent evolutionary innovation. While they are present in bony fish, lobe-finned fish, and tetrapods, they are not found in cartilaginous fish. Thus, bitter taste receptors must have first appeared in an aquatic species around 430 million years ago. This makes them the most recent of the chemosensory receptor families in vertebrates. The origins of the receptors involved in taste perception in animals are the subject of much speculation. Similarities between the evolution of vascular plants and the Cambrian explosion in animals suggest that the origin of these receptors may be linked to an important transition in plant to herbivore interactions. However, there is also evidence that these receptors play other roles, such as detecting nutrients in the gut, suggesting that their function as taste receptors may have emerged later. The bitter taste receptor family has diversified within and between taxa, with repertoires of bitter taste receptor genes varying in size across species genomes. Studies show that selective pressures on taste perception represent the most obvious potential driver of diversification of these genes. Fish have different numbers of genes, ranging from 1 in Amazon molly to 74 in coelacanth, but most fish have an intermediate number of genes. Variation in the number of bitter taste receptor genes occurs in different groups of animals, such as fish, amphibians, and saurians. Caecilian amphibians, which include limbless amphibians, have the lowest number of genes, while anurans, which include frogs and toads, have the highest number among all recorded vertebrates, probably reflecting their adaptation to variable lifestyles and environments. In fact, most frogs and toads inhabit both aquatic and terrestrial niches, which necessarily contain a greater variety of toxic substances. Consequently, their ecological needs should encompass very different requirements for their gustatory systems. For example, aquatic factors such as pH have been shown to influence the divergence of taste receptor genes. Most frogs feed on worms, insects, and other small arthropods, which contain more potentially toxic substances. It is known that insects often sequester toxic compounds from plants for their own defense, suggesting that anurans may benefit from the ability to detect dangerous prey. While caecilians are animals adapted to life underground, living in soil layers rich in organic matter, they build tunnels to get around. All caecilians are carnivores, feeding on earthworms, plathelminths, arthropods, frog eggs, and tadpoles. Compared with frogs, caecilians are less sensitive to bitter taste, as animal tissues contain fewer toxic substances. Consequently, we hypothesize that the lifestyle, ecological, and dietary complexity of amphibians raise their evolutionary pressure for a wide variety of taste receptor genes. Saurians, which include crocodiles, lizards, snakes, and birds, also show variation in the number of these genes. Snakes have low numbers, while crocodiles, turtles, and lizards have higher numbers, similar to many fish. Repertoires of bitter taste receptor genes in birds and mammals show great diversity. Penguins and cetaceans have few or no functional genes, while birds such as zebrafinch and white-throated sparrow have several. Primates, including man's closest relatives, have an average number of genes, with the gibbon having the fewest and the chimpanzee the most. Although toxins are a threat to all herbivorous animals, not all species are equally exposed. This variability is reflected in the size of genetic repertoires, which is related to the proportion of plant-based diets for each species. This trend is particularly marked with herbivores having larger repertoires than carnivores. The most extreme example of this variation is found in obligate carnivores such as whales, penguins, and pinnipeds, which swallow their food without chewing, resulting in a total absence of certain genes linked to toxic detection. Around 7 to 8 million years ago, humans had one last common ancestor with other great apes. If we look at the current eating habits of great apes, we can deduce the diet of our common ancestor. It seems that this species was omnivorous, feeding mainly on tropical fruits, and also included leaves and insects in its diet. Chimpanzees, our closest relatives, rely heavily on fruit for their caloric intake, with a small proportion of their diet coming from animals such as other apes and insects. Between 4.4 and 2.3 million years ago, human ancestors left the forest for the savanna and other ecosystems, and their food repertoire expanded considerably. Thus, over time, early hominids gradually abandoned the eating habits of forest monkeys and increased their ability to obtain a variety of nutrients in order to adopt a more diversified outdoor diet. Despite this dietary evolution, our need for fruit and our attraction to sugars and acids, shared with other great apes, remained intact. From a nutritional point of view, fruit is attractive because of its sugar content, which provides intrinsic satisfaction, as well as vitamin C, which is essential for life support in hominoids. The question of why humans are sensitive to acidic flavors and even inclined to prefer acidity has sparked debate in the scientific community. Signals from acidic substances do not provide much nutritional value, with a notable exception of vitamin C. However, this exception is significant because, unlike most mammals, monkeys and apes are unable to synthesize vitamin C due to the loss of a functional version of the gene encoding gluconolactone oxidase. The common ancestor of arthropods that suffered this enzymatic loss probably had to rely on a sufficiently rich source of ascorbic acid from fruits and other plants for the enzyme to become useless. It is plausible that the acidic taste played a necessary role as a guide to identifying vitamin C-rich fruit. In addition, the combination of acids with sugars could also have been used to assess the degree of ripeness of the fruit, using mixtures of sweet and sour flavors. Thus, acids are not stimuli for which we have evolved to react in isolation, but rather elements we have experienced in the context of the sugars present in fruit. As a result, sweet and sour flavors are perceived as synergistic in the fruit aroma palate. In addition, acid and sour taste act as indicators of fermentation processes which human beings around the world seek out and consume. Wild apes depend mainly on forest plants, especially young leaves, for most of their daily protein intake. Nevertheless, early hominids and modern humans have shown a preference for consuming slightly more digestible protein sources, such as meat. Although umami taste is not immediately apparent in fresh meats, it develops significantly in aged or cooked meats. Consequently, it seems that chimpanzees do not possess a gustatory system specifically dedicated to detecting the taste of glutamate or ribonucleotides, even though they can perceive these stimuli. The characteristic umami taste of hydrolyzed protein is mainly carried by glutamate and ribonucleotides. Humans have developed a preference for glutamate, ribonucleotides, and umami taste, probably as signals indicating more easily digestible proteins in lightly aged or cooked meats. Indeed, there is ample fossil evidence to suggest that culinary practices predate the advent of modern man. Humans the world over take advantage of a wide variety of plant and animal products that undergo fermentation. Our strong attraction to the taste of free amino acids and ribonucleotides may stem from a tendency to consume fermented foods, including lightly aged and/or cooked meats. This category of food would bring various benefits to the survival of our species. Fermentation facilitates access to macro and micronutrients while providing probiotic bacteria. Although it has been assumed that the pleasant taste of glutamate and ribonucleotides acts as an indicator of protein, many protein-rich foods are not particularly tasty or umami when fresh. It's the process of fermenting or aging these foods that releases the umami, savory taste of protein. Thus, our attraction to amino acids, particularly glutamate, as well as salty taste, may have emerged from our desire for fermented foods, taking advantage of the nutritional improvements and benefits of probiotic bacteria for our species. Humans stand out from other mammals thanks to a unique phenomenon, a large-scale polymorphism of the salivary amylase gene. This change in our genetics could be the result of our past history linked to the consumption of starch-rich tubers, and could have occurred around 100,000 to 200,000 years ago. The number of copies of the salivary amylase gene present in our genome directly influences the production of amylase in our saliva. This enzyme in high quantities and efficiency has the ability to rapidly transform a cooked starch, such as a thick pudding, from a semi-solid to a liquid state in a matter of moments in our mouths. The resulting sugar concentrations can produce sufficient substances such as maltose, which activate brain regions linked to taste and reward. In short, it seems that the human mouth harbors a taste receptor that responds to glucose polymers without necessarily inducing a conscious taste sensation. This receptor can be stimulated by the products of starch decomposition in the mouth, especially in the presence of high levels of salivary amylase. Human beings have a natural tendency to seek out the taste of salt in food, We find the taste of moderate concentrations of salt very appealing, as do other omnivores. But high levels of salt are considered unpalatable to us, as they disrupt the osmotic balance of our body fluids. Salt is a widely used ingredient in food preparation throughout the world, and individuals from different cultures consume roughly the same amount of salt every day. This similarity in salt consumption across cultures suggests that our salt intake is regulated by intrinsic biological factors. A carnivorous animal absorbs salt at every meal, while a herbivore can easily run out of sodium, triggering a desire for salt, which translates into a search for natural licks. An omnivore's exposure to dietary salt would therefore logically be intermediate between that of carnivores and herbivores. It should be noted, however, that humans lose salt through perspiration, which may explain why we have a slightly higher salt intake preference than other omnivores. Since our earliest prehistoric ancestors, the sense of touch has been central to our survival and reproduction. What we touch can have a direct impact on our ability to survive and reproduce. It may be a predator, prey, or parasite, a parent, lover or child, shelter or danger, a tool or food source, the ground beneath our feet, or the branch we use to swing. It's also the first sense to develop, with some fetuses responding to touch in the womb after just 8 weeks. Evolution has created a tactile system made up of different sensors and receptors, which together detect the qualities of what touches our body and what our body touches, such as weight, temperature, and texture, so that we can deduce useful information. Many animals, including some coelantherates, annelid worms, insects, and many other arthropods, birds, and mammals, have hairs or capillary projections that are richly supplied with nerves and serve to indicate to the animal that it is in contact with an object. These hairs may have been specially modified, for example, vibrissae or whiskers in certain areas of the body, such as the face or toes, to provide more sensitive discrimination between stimuli. A second type of tactile receptor, the subcutaneous receptor, is found in the skin. Receptors of this type are found in almost all animals and may consist of free nerve endings or complex corpuscles. Unicellular organisms already possess the capacity for directional detection of tactile stimuli, as can be seen in ciliates such as paramecia, which move away from the stimulus source following mechanical disruption of their outer membrane. Sponges, the simplest multicellular animals, do not possess neurons, but they do have the capacity to respond to variations in water flow and pressure in response to the movement of their immobile cilia. However, the evolution of neural conduction has led to a radical change in the ability to respond rapidly and flexibly to tactile stimuli. Cnidarians such as jellyfish and hydra, despite their relatively simple nervous systems, can exhibit coordinated patterns of motor response to sensory stimuli, and many have a rich capacity to respond to touch. For example, hydra nematocytes are structures that provide a good model for understanding the mechanoreceptors of more complex invertebrates. In jellyfish, groups of hair cells called tactile combs regulate complex behaviors, including escape, feeding, and locomotion. The advantages of sensitivity to mechanical stimuli provided by hair-like structures may have encouraged their convergent evolution in several animal lineages. For example, the ciliated cells of jellyfish appear to be sufficiently different from those of vertebrates that a common origin for both is unlikely. Studies into the molecular basis of mechanosensation in different classes of animals also suggest that the cellular mechanisms required for tactile detection may have evolved several times. All animals with a central nervous system respond to touch. The nematode, Sanor habditus elegans, is an important model organism in neuroscience. It is the first animal system in which the entire neural network involved in tactile behavior has been identified. This worm has mechanosensory cells located in the integument beneath the cuticle, linked to 6 sensory neurons, 10 interneurons, and 69 motor neurons. In larger, probably more advanced animals, touch-induced behavior becomes more versatile and complex. The leech has developed a central nervous system with a succession of ganglia for each segment. Each ganglion houses 3 groups of mechanosensory neurons, all of which respond to mechanical stimulation of the body, but with different response thresholds. Tactile cells are the most reactive and respond to the gentle touch of the body wall. Pressure cells respond to more intense tactile stimuli, while nociceptive cells are sensitive to very powerful and potentially harmful stimuli. In arthropods, mechanoreceptors are located in strategic areas of the body where movement is most likely to occur. In proprioception, these are the joints, muscles, and tendons. In contact, they are the surface areas where contact with external objects is most likely. Arthropods exploit these strategically important locations in 2 ways. On the one hand, they use tufts of hairs called capillary fields, or capillary plates, to detect movement between adjacent body segments through proprioception. On the other hand, to detect contact, they use similar hairs but located in different parts of the body, with a particularly high concentration where contact is most likely and/or most important to detect. Spiders and insects also use hair sensilla for proprioception and consequently for active touch detection, where the active movement of a limb must be monitored. Among today's arthropods, only myriapods, crustaceans, and insects have specialized touch sensory structures, the so-called antennae. These sensory organs are equipped with a particularly large number of sensillae, displaying a density far greater than that found on most other body regions. Crustaceans have 2 pairs of antennae, while insects have only 1 pair. The generally accepted hypothesis is that over the course of evolution, insects are likely to have lost their second pair of antennae, meaning that the insect antenna shares a common evolutionary origin with that of crustaceans. Fish and amphibians have mechanoreceptive organs called neuromasts, which are arranged in rows on their body, head, and tail. These neuromasts may be present autonomously on their skin or inside fluid-filled channels. The main sensory component of each neuromast is a group of hair cells that respond to hydrodynamic stimuli caused by water movement or variations in water pressure. In this way, the lateral lines provide a sophisticated sensory system for detecting the distant origins of water movement, such as predator or prey behavior, water currents, and the topographical features of the underwater environment. Mammalian skin evolved from the integument of earlier vertebrates in a complex evolutionary process that is still only partially identified. What is clear is that the first mammals, or perhaps their reptilian, therapsid ancestors, redeveloped hair as an adaptation of the outer epidermal layers of their integument. While dense hair clearly has a thermoregulatory function, the evolution of hair in mammals cannot be attributed solely to the goal of regulating body temperature. On the contrary, it is very likely that the first hairs were primarily used for tactile functions, and their subsequent evolution gradually gave them a secondary role as insulators. The main sensory function of hairs persists in vibrissae, also known as tactile hairs, present in all therian mammals, such as marsupials and placentals, with the exception of human beings. In cases such as the naked mole rat, a species that has lost its entire coat, tactile hairs have been maintained throughout the body because of their importance to the sense of touch. Many mammals have evolved to have areas of hairless skin, also known as glabrous skin. In humans, these areas include the skin of the lips, hands, fingertips, and soles of the feet. These are the parts of the body that play a key role in physical interactions with the outside world, and where tactile precision is particularly important. So it's not surprising that hairless skin is characterized by a high density of mechanosensory receptors that are extremely sensitive to delicate touch, which could contribute to the effective or social tactile capacity that could be specific to mammals. Only mammalian species have elongated facial vibrissae, also known as vibraceae, which extend outwards and forwards from the animal's snout. They form a tactile sensory network that envelops the animal's head. Species without vibraceae, or in which they were less apparent, were generally the most evolved representatives of their order. Consequently, having facial vibraceae was an ancestral characteristic of mammals. Many terrestrial mammals, especially those that are nocturnal, live in low-light environments or climb trees, and have evolved highly specialized tactile sensing systems via vibraceae. Research has focused on a variety of these animals, including rodents such as the rat, mouse, and golden hamster, as well as insectivores such as the Etruscan shrew and the Brazilian short-tailed marsupial possum. In some seal species, vibraceae have adapted to detect disturbances left in the water by fish. By measuring these hydrodynamic flow fields, these animals are able to track and capture fast-moving prey in total darkness. Deep-sea foraging pinnipeds, such as the bearded seal and walrus, have evolved a highly specific vibrissae adaptation designed to optimize their efficient search for shellfish and other invertebrates in the muddy layers of the seabed. The abundant, dense, and mobile vibraceae of these animals combine to form a kind of sensitive, tactile rate that enables them to detect and ingest their prey at exceptionally fast speeds. Manatees have vibraceae distributed throughout their bodies to compensate for the limited visual information available in their natural environment. The manatee's ability to move efficiently through rough waters suggests an overall body ability to perceive hydrodynamic signals to the sensitivity of its vibraceae, which has interesting parallels with the lateral line of fish. These adaptations underline the importance of vibrissae touch detection in the ecology of many aquatic mammals. Human fetuses begin to sense their environment through touch as early as the 8th week of gestation, contributing to their body awareness. After birth, the mouth evolves to become the primary means of discovering surrounding objects. But this haptic focus shifts to the hands as we grow older. It's not surprising that humans lost their facial whiskers over the course of evolution, as our hands took over as tools for tactile exploration of the immediate environment, while also enabling us to manipulate with skill and precision. Unicellular and multicellular organisms have developed different strategies to ensure their survival and communication with their environment. These strategies include molecular, anatomical, and physiological systems that enable them to detect and respond to chemical signals from other cells or organisms. These communication mechanisms are the result of organisms' ongoing adaptation to environmental factors and biotic interactions. Over time, organisms have developed adapted receptors that enable them to detect and respond to specific signals.